back to another session of biochemistry classes. In the last class, we finished with the other aromatic amino acid metabolisms, that is the phenylalanine and tyrosine. So today's class we'll go on to the tryptophan metabolism. So if you remember from the chemistry, tryptophan is a both glucogenic amino acid and ketogenic amino acid. It also helps in synthesis of certain proteins and also indole acetic acid. So tryptophan along with it helps in the synthesis of certain important products or compounds like the coenzyme of niacin, that is NAD, NADP, serotonin, melatonin and 5-hydroxyindole acetic acid that gets excreted in the urine. Okay, so what is tryptophan? It is an aromatic indole ring containing essential glucogenic and ketogenic amino acid. So it's a precursor for the synthesis of important compounds like NAD and NADP, which are the coenzymes of niacin, serotonin and melatonin. So tryptophan metabolism, we can classify it into two main pathways. One is the kynurenine or the kynurenine anthranilate pathway, and then the serotonin pathway. So we'll go on to the first pathway, the kynurenine anthranilate pathway. So here tryptophan, in the presence of tryptophan oxygenase, gets oxidized in the presence of an iron molecule to form L-formyl kynurenine. Now this L-formyl kynurenine, in the presence of kynurenine formylase enzyme, utilizes one water molecule and fumarate is given out to form L-kynurenine. And this kynurenine, in the presence of kynurenine hydroxylase enzyme, the presence of NADPH and H, gets oxidized to form 3-hydroxykynurenine. This 3-hydroxykynurenine, in the presence of kynurenase enzyme, which requires pyridoxal phosphate as the coenzyme, converts water molecule here to form alanine, and this alanine enters into the glucogenic pathway. Okay, now this 3-hydroxyanthranilate that is formed by kynurenase enzyme gets converted by several steps to form the acetyl CoA, and acetyl CoA enters into the ketogenic pathway. Okay, now 3-hydroxyanthranilate, by several steps it gets converted to quinolate, and this on oxidation gives niacin and leads to the synthesis of the coenzyme, that is NAD. Okay, so when there is deficiency of vitamin B6, what happens? This 3-hydroxykynurenine will not get converted to 3-hydroxyanthranilate. Now this 3-hydroxykynurenine gets converted to xanthronate and gets excreted in the urine. Okay, so this is whenever there is deficiency of vitamin B6. Okay, so this is the breakdown pathway or the catabolism of tryptophan to give the alanine that enters into the glucogenic pathway, acetyl CoA that enters into the ketogenic pathway, and also it leads to the synthesis of niacin and the coenzyme form of niacin, that is NAD. Okay, so this is the kynurenine pathway. So we can just have a brief discussion about the kynurenine pathway. So what is it? The initial reaction is a oxidation of tryptophan to N-formyl kynurenine, which is catalyzed by tryptophan pyrrolase, which is having a feedback inhibited by NADH or NADPH. Okay, so there what happens? Further removal of the formyl group by kynurenine formylase enzyme to form kynurenine. Now further the kynurenine gets converted to 3-hydroxykynurenine by kynurenine hydroxylase enzyme. Now what happens then? The 3-hydroxykynurenine gets converted to 3-hydroxyanthranilate and alanine by kynurenase enzyme, and this alanine enters into the glucogenic pathway. So this requires PLP as a coenzyme. So any deficiency of vitamin B6 will result in failure of catabolism of hydroxykynurenine. It forms xanthronate and it gets excreted in the urine. Now 3-hydroxyanthranilate undergoes decarboxylation to get converted to form the niacin, and this converts to NAD and NADP or certain other 3-hydroxyanthranate by multiple steps gets converted to acetyl CoA, and this acetyl CoA enters into the ketogenic pathway. Okay, so this is the kynurenine pathway. Now we'll go on to the nicotinic acid, how it is formed. So what is it? 3-hydroxyanthranilic acid gets converted to NAD by quinolate phosphoribosyl transferase enzyme. This is the rate limiting step. So 60 milligrams of tryptophan will give around 1 milligram of niacin. So whenever there is deficiency, pellagra is caused. So when now these pellagra is seen mostly in people who take staple diet as maize, because maize has deficiency of tryptophan. Okay, so this is about the nicotinic acid pathway. So next we'll go on to the serotonin pathway. So what is it here? Tryptophan, in the presence of tryptophan hydroxylase enzyme, this requires tetrahydrobiopterin, gets converted to 5-hydroxytryptophan. This 5-hydroxytryptophan by decarboxylation gets converted to 5-hydroxytryptamine, that is nothing but serotonin. Now here an acetyl group gets attached to the serotonin by acetyl transferase enzyme, and it forms N-acetyl serotonin. This N-acetyl serotonin, there is an attachment of another methyl group by methyl transferase enzyme, which is donated by S-adenosyl methionine or SAM to get converted to N-acetyl 5-methoxy-serotonin, which is nothing but melatonin. Okay. Now serotonin further, in the presence of a monoamino oxidase enzyme, gets converted to 5-hydroxy-indole acetic acid and gets excreted in the urine. Okay, so this is the serotonin metabolic pathway where tryptophan gets broken down or catabolized to form melatonin and 5-hydroxy-acetic acid, and also serotonin is synthesized. Okay, next we'll just go into the deep discussion about what is the serotonin pathway. So tryptophan is first oxidized to 5-hydroxytryptophan by tryptophan hydroxylase, which requires tetrahydrobiopterin as a cofactor. Now 5-hydroxytryptophan undergoes decarboxylation to form the serotonin of 5-hydroxytryptamine, which requires PLP. Now after this, an acetyl group of the serotonin is added, followed by methyl group in the pineal glands to form the hormone melatonin. Okay, so this is the serotonin pathway. So next we'll go on to what are the important compounds that are synthesized or derived from tryptophan. You have the neurotransmitter, that is serotonin, and then you have the hormone melatonin, and also the vitamin niacin, that is B6. Okay, so first we'll go to serotonin. What is the function of serotonin? It is synthesized by the neurons, the pineal glands, and by the argentifying tissues of the abdominal cavity. So it has a neurotransmitter property. It is a potent vasoconstrictor and stimulator of the smooth muscle, and also construction of the smooth muscle. So oxidative deamination of serotonin by the enzyme monoamino oxidase leads to the formation of 5-hydroxy-indole acetic acid, okay, which is excreted in the urine. So serotonin is a neurotransmitter and it performs a number of functions. So it is a powerful vasoconstrictor and it results in smooth muscle contraction in the bronchioles and in the arterioles. Also it is helpful in regulation of the cerebral activity, and it controls a behavioral pattern, sleep, blood pressure, and also the body temperature. And serotonin also releases the peptide hormones from the gastrointestinal tract, and it is also required for the motility of the gastrointestinal tract. And serotonin is a stimulator of the brain activity, and deficiency of this causes depression, and decreased levels are seen in psychosis patients. Okay, this is the important or the functions of serotonin. So we'll go on to another um, uh, carcinoid tumor, which we call it as the argentophenomas. Okay, so when we talk about this, it can come as a short note or a viva question for you. So what is it? It is a serotonin secreting neoplasm, develops from the enterochromaffin cells. Okay, this is distributed in the GIT, biliary tract, and also in the gallbladder, pancreatic duct, and the bronchial tree. So mainly where there is overproduction of serotonin, it is mostly seen, and up to 60% of tryptophan is converted into serotonin. So because it is involved with the tryptophan metabolism, the patients will have pellagra-like signs and symptoms. Okay, so what are the clinical features of carcinoid tumor? Prominent you have see is the prominent flushing, bronchial constriction, then you have the diarrhea, then cardiac lesions and associated with heart failure, increased excretion of the urinary 5-hydroxyindole acetic acid, right? So how do you diagnose this condition? You do the measurement of the urinary excretion of 5-HIAA concentration. So what do you advise to a patient when you have to do this test? Prior to doing this test, you advise to the patient not to take any medications for 72 hours, excluding certain dietary sources like avocado, banana, pineapple, tomato, and also certain cough syrups that contain indomethacin. Okay, so this is going to be the first advice, and then we collect the random fresh urine sample is taken, and this 5-HIAA is estimated. So the normal levels are around 2 to 7 milligrams per day. Increased levels are seen in carcinoid tumors in the gut or in the bronchus, tropical sprue, Whipple's disease, oat cell carcinoma of the bronchus. Levels more than 25 milligrams per day is diagnostic of carcinoid tumors. Anything more than 350 milligrams per day is indicative of metastatic tumors. Okay, so decreased levels are also seen in depression, phenylketonuria, and Haffner's disease. So this is about the carcinoid tumor. Next we'll go on to the functions of melatonin. So melatonin is involved in circadian rhythm or the diurnal variation, that is the 24-hour cyclic process of the body, plays an important role in the sleep and the wake pattern of a person. So melatonin inhibits the production of melanocyte stimulating hormone and the adrenal corticotropic hormone. It also has inhibitory effect on the ovarian function. Melatonin is also a neurotransmitter. Okay, so whenever we talk about serotonin and melatonin, you have to talk about also the metabolic disorders of tryptophan, that is the Haffner's disease. Okay, so what is it? It is impaired intestine and renal transport of tryptophan and other neutral amino acids. Okay, there is decreased tryptophan availability for niacin synthesis, and most of it there are signs of pellagra like signs and symptoms, right? So what are the clinical features? There is photosensitivity and red scaling, okay, which resemble that of pellagra. There is cerebral ataxia, headache, muscle discomfort, psychological symptoms are also seen, and then there is generalized amino acid urea. Okay, so this severity of this Haffner's disease is less in the adult life, right? So what are the diagnostic tests that can be done to detect the Haffner's diseases? The urinary excretion of indole acetic acid and also indole pyruvic acid and the other indole derivatives can be looked into the urine. Okay, any deduction of elevated amino acids in the excretion, okay, can be seen. Then what do? You take the urine specimen and you do a chromatography pattern and deduct what is the excretion in the urine, right? And then you can also do a molecular genetic testing that can confirm the Haffner's disease, right? So how do you manage? What is the treatment for Haffner's disease? You give a oral nicotinic acid supplement up to 300 milligrams per day is given, okay, and also rich protein diet is given to these patients. But what is the most important thing is here the pellagra like symptoms is always corrected because of the giving the nicotinic acid supplements. But what happens here, the amino acid urea is not altered, so there is excretion of amino acid in the urine, okay. So this is the treatment for Haffner's disease. So this finishes with tryptophan metabolism. So most of the time in tryptophan metabolism, the catabolism of tryptophan comes as a question, okay. So then you will have to write both the kynurenine pathway and also the serotonin pathway, okay. And then you will get most of the time even the HIAA can come as a question, or even Haffner's disease can come as a question, and also a viva question, okay. So this finishes with tryptophan metabolism. In the next class we'll go on to another metabolic pathway. Thank you and have a nice day.